Hello, and welcome to the Crown Bioscience, a novel approach to overcoming limitations of T-cell immunotherapies webinar. Your presenters will be Dr. Maloy Ghosh from Zumutor Biologics and Dr. Nataliia Beztsinna from Crown Bioscience. Before we begin, there are just a few notes about the broadcast. Your audio will be placed in listen-only mode during the duration of the presentation. If you have any questions during the webinar, please type them in the question and answer box, and we will try to answer all questions before the end of the Q&A session. You will only be able to chat with the presenter, not other participants. And we expect the presentation and engaging Q&A session that follows to last a little over an hour, and hope you will stay with us for the entire session. And now let's get started with Dr. Ghosh from Zumutor.
Hello, everybody. My name is Dr. Maloy Ghosh. I am CSO of Zumutor Biologics, and I'm presenting our work at Zumutor Biologics, where we have used this unique platform from Crown Bio, and quite an interesting data set came out of it, and this looks like something interesting as a molecule and pathway, and of course the platform that we are using for this particular work. So I will divide my talk into two parts. The first part, I will have a small introduction about Zumutor Biologics and the work that we do on novel biologics development space. And then I will get into some of the data that we have generated, especially using the Crown Bio platform. So Zumutor Biologics is a company based out of Bangalore, India, and Boston at Cambridge. So this is our team, myself, Maloy, and Kavita is our CEO. And the core team also have our CFO, Mr. Anirban Bhattacharya. So all three of us know each other for a long time, and this company is almost 10 years old. We started as a startup when our interest was to develop novel monoclonal antibody, not the biosimilar ones, but drugs which could be next generation drug. So when we started our work, we had an idea to develop novel molecules. Now, like in small chemical molecules, drug molecules, for larger biologics, developing a novel molecule is not so straightforward. So we started with a couple of platforms that we thought will provide us this ability to generate new molecules. One of them is our growth engine. Essentially, we call it enabler. It's a monoclonal antibody library platform. Very, very large monoclonal antibody library. Genes are cloned into this library, both naive and synthetic molecules. And then we also have a very strong and robust selection platform, which is a combination of phage and yeast surface display. And we also have another platform where we have improved the molecules ADCC function. Some of the molecules might require this for their function. And the idea was from the very beginning, utilize these two platforms to develop new molecule. We are quite successful in establishing that. The enabler platform starts with, as I said, very, very large number of monoclonal antibody genes. They are harvested from healthy humans, multiple of them. And then we go through a series of selection process. I will not go to the details of this. We have published IPs on them. Many of those IPs are granted in US, in Europe, in Japan, in Canada, in majority of the geographies where this kind of drug molecules are have a huge market potential. We utilize the fudge surface display and yeast surface display as a mechanism to select the molecule of our choice. And this process is quite well established now. We take close to 4 to 6 months to develop our target candidates, and then we develop those molecules. And it can be done for any target antigen. As a company, we have a very strong focus on a particular area of research, which is known as, okay, before going there, these are the different antibody platforms that we have. We are not a service company. We utilize these platforms for identifying our pipeline molecules. But again, we do collaborate with other companies, institutes, wherever the project is interesting. And there is a challenge involved where these technologies can be used to identify new molecules. We do collaborate on a very selective basis. These particular libraries are very well studied now. We have a large amount of data generated on the composition of the library. So this particular graph shows in antibody, as you know, heavy chain CDR3 is the main CDR which interacts with the antigen. And you can see in this curve, there are 3 different things we are trying to say. First, the color indicates the type of amino acid. There are multiple amino acids with their single letter identifier. The height of each bar represents the percentage occurrence of that particular amino acid, whatever the color is, and the position indicates where in the CDR they are. For example, this one is 28 position in the amino acid number in the CDR1. Similarly, if you can look at CDR, heavy chain CDR3, this is the region which is the loop structure, highly variable. Many amino acids are present at different percentage, although some are more prevalent than the others. And these are typical, prevalence rate that we have seen and others also have seen. Our library is also, when we tested the sequences, has a large number of germline family genes, which is another hallmark of understanding antibody diversity. We actually have covered more than 80% of all the germline families reported so far. So each one of them are actually a particular family, germline family of the antibody gene, and we can identify presence of them throughout our library when we did the sequence analysis of CDS. The V genes. We utilize this library now. As we have generated this library, we came up with an idea that where we should utilize these libraries for product development. So we identified a particular niche in oncology, more specifically saying immuno-oncology, where we thought that newer molecules have an immense potential in terms of helping the patients. Now, if you look at immuno-oncology field as such, and I'm talking about when we started working on this in 2015-16 timeframe, majority of the targets people were working on were derived from T-cell based pathways, mostly somewhere or other linked with PD-1, PD-L1 interaction. And these molecules were, obviously anti-PD-1 and anti-PD-L1 are immensely potential molecule. They have proven so. Nobel Prize has been given on them. And but the next set of molecules, which many of them are targeting the similar pathways. So as a strategic thoughts, we shifted from T-cell immunotherapy to a different immune cells, which we thought are quite important in protecting us from many infections, including cancer, and the evidences were coming out by that time known as natural killer cells. So we looked at various markers which are present on natural killer cells and driving the process of tumor escape through natural killer cell mediated pathways. And we decided to land up on a particular marker on the surface of NK cell, and which interacts with the compound X, what we call now as compound X on tumor cell. So this particular target on tumor cell is quite widespread, present itself on multiple tumors. Majority of them are solid tumors. So we were interested in that particular domain using innate immune system, an immune cell like NK cell, and targeting tumors, not primarily the lymphoma and leukemia set of cancers, but mostly the solid tumors, the tumors which has less, the drugs are not so much effective on cancers like this. So that's how we selected our target and then utilized our platform to develop the antibody, which we call novel compound X. So this is an antibody came out from our platform. It's an human IgG1 molecule, and it disrupts this interaction specifically. The data that we have generated so far suggests this specific disruption of this interaction between compound X, or the target X, and its receptor on the NK cell leads to NK cell activation and various other mechanisms through which tumor can be controlled. So we developed that molecule, and as a company, we built up a pipeline primarily targeting NK cell. There are 2 more molecules in our pipeline. They are undisclosed. The target is undisclosed so far. They are early in development in the discovery preclinical stage. The molecule that we are going to talk about today, we have generated very strong preclinical data, including the platform that we have used from Crown Bio. And finally, last year in August, we received the FDA the IND approval of this molecule. Right now, this molecule is ready to initiate patient dosing in clinic. We are going ahead with 5 different sites in US for phase 1 clinical trial. So I'll go to the next set of slides where we will talk more about this compound X and some of its function that we think are quite interesting to observe. So to give you an overview of what we have done so far, because this is a new molecule, new pathway, we had to deal with a large number of experiments where it's a first in class, by the way. So a large number of experiments were run to establish the mode of function or mode of action of this molecule. We filed our IP. The IP was granted in US last year again in 2023. And we are going to get the grants in Europe and other countries. It's a global IP has been followed here. And we have now data which suggest there is a significant monotherapy potential for this molecule, which we call compound X. We have shown that in vitro experiment. We have shown that in mice model, which are humanized mice, and where we have seen a significant infiltration of immune cell, a hallmark of immuno-oncotherapy, and which is quite interesting when we ran the Crown Bio platform, where we have used 2 different disease platforms, essentially in lung and in bladder cancer. We have seen similar type of immune cell infiltration, which is quite interesting to see. And this molecule also has been tested as a monotherapy as well as in combination of an very well-known anti-PD-1 antibody, which is pembrolizumab. The molecule shows very clean pharmacology and toxicology profile in monkeys, in cyno monkeys that we have tested. And finally, we got the IND grant in in last year. By March, we will inject. The first infusion will be done by March. So as I was telling, our primary interest is in solid cancers, various solid cancers, where this antigen is well expressed in the tumors. Just to look at data which is already present in literature. So this is the analysis of TCGA database, the transcriptome analysis, and many of the cancer across these 33 different cancers that we have looked at have shown higher transcript level in the TME compared to adjacent normal. So this is interesting because this kind of gives us a very nice broad spectrum understanding how this antigen expression could help us choosing the tumors that are of requirement, where the standard of care, the current standard of care is not so effective. So when you work with any of these immuno-oncology antibody, the primary function of this particular drug molecule is killing the tumor cell. So that is the first thing you looked at. So we looked at two different tumor cell types. One of them is PC3, which is a very, very aggressive prostate cancer cell line. We looked at how in presence of our antibody, the tumor cells die. So this is a flow cytometer experiment, very simple, where we have added our antibody and monitored the cell death. So this particular quadrant represents the dead cells through a dye. And you can see a majority of the tumor cells compared to no antibody control are dead here. So we had proper controls like an antibody, which is a human IgG, and compared that against our drug of interest, which is compound X. Similarly, we looked at this particular tumor cell types, which is known as K562. So the difference between these two are interesting. These cells are having MHCs on their surface, and these cells do not have any MHC. So because this molecule is working through NK cell pathway, NK cells do not require MHCs to recognize the tumor cell, and we do see nice tumor cell death in this case as well. So going forward, our next objective was, well, the immune cells, what is the function of immune cells in this context, correct? The tumor cells are dying in presence of our antibody and healthy immune cells. So how the immune cells are helping the tumors, causing the tumor death. So one of the very known, well-established pathway is the IFN gamma pathway, through which immune cells do kill the tumor cells, and we do see a significant increase in IFN gamma production in presence of our antibody. Then we followed up this experiment with many different cell types. We have seen both NK cells as well as CD8 T cells in presence of this particular antibody produces IFN gamma intracellularly, and that IFN gamma is responsible for tumor cell death. We looked at other activation signals or cell death signal. For example, CD107 is a typical marker that you look at when you see that the immune cells are killing the tumor cells, and we do see a significant overexpression of CD107 as well. Going next part, it's we thought that it would be nice to have a great visualization of how the tumor cells are getting killed. So we ran a very nice imaging study where we carried out confocal imaging as well as the imaging at different time points. So this is one of those experimental data where the tumor cells are labeled with a dye, which is very nice green, large tumor cells, very happy in presence of the immune cells, the red cells. Now, when you add antibody in the mix, then what happens? These immune cells come in contact with these tumor cells, they start killing them. Once they start killing them, the membrane is now compromised. There is a color present in the media, which then able to enter the cell and the cell turns blue. So this is a nice way of representing the tumor cell death and visualize how many tumor cells are dying. So we generated nice image out of these experiments. We ran these experiments with total PBMCs. We ran these experiments with isolated NK cells and various other immune cells. And we do see dose-dependent increase. As we increase the dose of our antibody, we see a corresponding increase in cell death. Very nice demonstration of the function of this molecule. The next set of experiment we looked at in mice, where this is an humanized mice, where we have created the tumor using PC3 cells. The tumor grows very, very fast. We inject our drug at particular days at a certain dose, and we do see a very nice reduction of tumor. Not only that, when we isolated these tumors at different time points and then looked at through immunohistochemistry, what is the change is happening within the tumor microenvironment, we do see a significant infiltration of not only NK cells, but also CD8 positive T cells. These are the immune cell infiltration data, which actually kind of directed us to study it in further detail. And then we came across this platform, which is from Crown Bio, where you can test this hypothesis clearly, correct? You can create the tumor, you can incubate the tumoroids in presence of drug, and that is what we have done to see how the human tumoroids actually work in presence of drug. So in this case, we have done 3 different treatment schedule. One is with our compound as a monotherapy. We used pembrolizumab as a standard of care monotherapy and a combination of the two. What we have done additionally here in this case, we have collected immune cell from the same patient. So it's autologous immune cell, labeled them with a dye, and then put them into the mix. So the idea was, can we see infiltration of this labeled immune cell in presence of our drug? And we tested that in 2 different sets of tumors from different patients, lung cancer patients and muscle invasive bladder cancer patient. And this data was quite useful when we submitted our application for US FDA IND application. And the reviewers actually appreciated the data, giving a very strong evidence of how a novel molecule is functioning in a human tumor system. So just a little bit of background, the samples, when you do this kind of analysis, you need to be very, very careful how the sample is collected, the type of sample. Not everything will work out. There's a very strong quality criteria is established within this platform, where only certain tumors with a certain outcome are allowed to go forward in presence of drug. Again, the patients are completely anonymous, and we collected the tissues only and then went ahead to do the experiments. We could save some amount of tissue, we could save some amount of soup for isolation and measuring the concentration of cytokines. You can do some amount of immunohistochemistry. But remember, these are tumoroids, so they're not really like tissues that you see in an actual tumor microenvironment. But this tissue or tumoroid sections represent very well, including the immune cells, the TME conditions. So we had several controls, no antibody control, super antigen, then human isotype controls, pembrolizumab and staurosporine, obviously. To understand how the tumor is behaving, we had monotherapy at different concentrations of our drug, combination therapy where pembrolizumab has a single dose and our drug was combined at 3 different dosage. And generally these experiments ran is within 6 days you collect the data. So this is how the tumorides look like at the beginning of the assay, and then you qualify. There is a quality check, which kind of, the plate is showing nice tumoroid formation, then only it goes forward. I'm showing a representative field here, how the tumors formed. And this is how the data is collected. So these are different treatments, and there are other treatments. I just presented 4 of them here just for comparison. And across is the sample. So this is one sample, second sample, third, fourth, fifth, like that. And the color that you see once the image is taken, it's completely digitized. So you can separate out the cells that you want, the immune cells, the tumor cells, the clumps, single cells, everything. So the data analysis is quite straightforward and very, very robust. This is the interesting part. So this is, I'm presenting the data in a qualitative manner across 5 different samples of lung cancer patients. And you can see the more yellowish the color is, more response you can see. Pembrolizumab by itself works only in 1 sample. The other 4 samples actually do not show any pembrolizumab response. On the other hand, when we put our drug, we do see response in other samples, varied response. And this is what is expected in any immunotherapy. The response is not equivalent in all patients. There is a lot other things which are responsible for the drug to work. But from this data, it is clear either in monotherapy or in combination therapy, both sets of samples actually have shown multiple samples responding in presence of our drug. So this is something quite exciting for us. 3 out of 5 we have seen responses, and this is something which we have taken forward in our clinical trial design. And lastly, I want to show you some of the images that we collected. These are again confocal imaging of the tumoroids. This is the control where the tumoroid structure is pretty big. These green cells are the immune cells which are labeled and put it in the mix. These are autologous immune cells. And you can see in presence of our drug, the tumoroid size has reduced significantly. The green cells are kind of inside the tumoroid structure, hallmark of reducing, how the tumoroid size is reducing. And in some cases, these tumoroid structures are almost gone. For example, in this particular field, we are kind of missing the large tumoroid structure. They're all almost gone or completely disintegrated, essentially suggesting this molecule by itself or in combination with an anti-PD-1 drug, which is the standard of care, correct, for lung cancer patients, work perfectly. And this is what we are going to test in our clinical trial now, which is going to begin next month. So to conclude, this is a new pathway. We do see innate immune system involvement in these molecules' work. We do see a very significantly high expression of the target antigen in multiple cancers, and the therapeutic usage of the molecule is really highly potential. The cytotoxicity mode of action we have established through multiple means, and large number of pathways actually has been interrogated here. We think that there is a significant collaboration of immune cells, NK cells, as well as T cells in terms of infiltration of tumor microenvironment and its function to reduce the tumor area. Ex vivo study, which is the model from Crown Bio, shows a significant reduction in monotherapy and combination therapy. And the molecule has a very clean pharmacology toxicology profile. We got our IND granted recently, and we're quite confident about the function of this molecule in the patients that we will recruit in this study. And we believe that many of these terminal patients will be benefited with a drug like this, where innate immune system is utilized in addition to the T cell based system, for example, in this case, pembrolizumab. So I'll stop here. You can connect me at Zumutor And obviously, if you want to share your questions through the webinar platform, I will appreciate that. Thank you very much, and you have a nice day.
And now we will transition to Dr. Beztsinna.
Hi, everyone. Thank you for joining this second part of the webinar, where I will show you more in detail what we can do in 3D ex vivo patient tissue platform here at Crown Bioscience, and hope to help you understand how we can better evaluate oncology drugs on patient tumors with preserved native tumor microenvironment. Here is a brief overview of my presentation today. I will show you the ex vivo patient tissue platform, or EVPT, assay principles, what we can do in the platform, and some example data for standard of care responses and also immuno-oncology drug testing. The idea behind this platform was to find a better translational model that will help to evaluate drugs with the very complex mechanisms of action. In particular, immuno-oncology drugs that require the presence of innate immune cells and, we know for immune checkpoint inhibitors, also exhausted immune cells in a sample. And as we know, there is no perfect model for this type of drugs, nor in vitro nor in vivo. So we tried to use this system to recapitulate the tumor microenvironment as close as possible ex vivo. In Crown Bio, we have different modalities that help to reconstitute tumor microenvironment. On one hand, we can use patient-derived organoids together with other cells, immune cells, fibroblasts that are isolated from healthy donors. This is what we call reconstituted tumor microenvironment assays. And on the other hand, we can use a fresh tumor sample that contains all the cells present in a human TME, and we can proceed from that sample directly. And this is what I'm going to show you today. So in a nutshell, this is the principle of how our EVPT platform works. We receive fresh patient tissue from the hospitals within 24 hours after it's been collected from the patient, and we process it right away. Our processing involves generation of tumor clusters. So we don't make single cells, but the clusters. Upon generation of these tumor clusters, also the cells that are native to the TME are released, including immune cells and the fibroblasts and other stromal components. This mixture of tumor clusters and other cells of the TME is then mixed with proprietary defined hydrogel and seeded in 384-well plate format. We seed on average from 50 to 300 tumor clusters per well in this format. And this allows us to test drugs directly on the same day after seeding in this 384-well plate format. We typically have 8 replicate wells per treatment condition, and with one patient sample we can test ranging from 15 to 25 different treatment conditions in this format. So as you can see, it's a medium throughput format. So once the tumor clusters are seeded and treated with the compounds, the assay is lasting from 6 to 7 days. It can be a bit longer, can be shorter, depends on the drug's mechanism of action. And this is an endpoint assay. So then the plates are fixed and stained and imaged in 3D using our high-content imaging system. Afterwards, we evaluate drug effects on the tumor clusters and immune cells using our proprietary phenotypic high-content image analysis platform. On this slide, you can see an example of a fresh patient tissue sample that has been just processed. Here on the left, you can observe tumor cluster and other cells from the TME in suspension. Once this sample is seeded in a hydrogel, we can observe a rich tumor microenvironment present in the sample, comprising from tumor clusters, cancer-associated fibroblasts, and also the little small cells here are immune cells. And if the same sample would be cultured long enough in 3D, after several passages, all the normal cells from the tumor microenvironment will disappear, and we will end up with a patient-derived organoid. So the complexity between the two systems is the main difference between the PDO and the fresh ex vivo tumor tissues. Also, when we were developing this platform, we were aiming to preserve the original patient biology of the tumor as much as possible, and our collaboration with the hospital pathologist has proven that our ex vivo 3D cultures are able to recapitulate the biology of the original patient tumor, as you can see here on the right. We work with multiple different cancer types, and it really depends on the supply and occurrence of certain cancer types and possibility of a surgery with certain tissues. So our main tissues are from surgical resected samples. We can also receive ascites or fluids from where we can also isolate metastatic tumor clusters, but mainly we work with surgeries. And what we call a standard offering means that these tissues were validated in a platform and we have a good supply of this material, and we can run assay in relatively short time frame for these cancer types. What we call customized offering here are the cancer types that were validated in the assay but are much more rare or more difficultly available samples. So the timelines can be longer, as for instance glioblastoma is extremely rare cancer type, as you know, or for pancreatic cancer we found that resections don't work really well. Ascites work much better, but this is much a rare occurrence as well. So for every project we discuss with our clients what would be the best indication for their drugs to test and how often we can get those samples in the pipeline. So here are all the capabilities of this platform. As I mentioned, we receive a fresh tumor resection that is later processed in a way to generate multicellular tumor clusters and liberate different cells from the tumor microenvironment. The sample needs to be processed fresh, and the amount of clinical information we receive at the time when the sample comes to us is really limited. So if we want to know more about the sample, we can perform additional sample characterization in-house. We can generate single-cell suspension and then perform FACS analysis on the different subtypes of the cells present in the TME. We can also generate a sample for immunohistochemistry from the processed tumor or a sample for sequencing. For the assays, we have two types of assays. One is the high-content imaging assay, which I already presented, and the main readouts for this assay would be tumor killing, immune cell proliferation, and immune cell infiltration if we work with autologous co-cultures, what Maloy showed in his presentation. We also have now a possibility of a scale-up assay, where we would use more material per well, and then this material would be further extracted from these wells, and we can again run FACS analysis on it and have slightly different readouts. In addition to the standard readouts, we can add extra readouts, which would be cytokine measurement that is possible from the supernatants of both high-content imaging and a scale-up assay. And also, we have validated a couple of antibodies for immunofluorescence staining to do directly in the assay plate. Here are some examples of the sample characterization that we've done in-house. These are done with non-small cell lung cancer samples. You can see here a Treg detection with either immunohistochemistry or FACS. And you can also see that in our high-content imaging plates, we have nicely preserved EpCAM-positive tumor clusters and CD3-positive T cells that are also clustering around these tumor fragments. Here are the example data on 4 cancer patients, samples from breast cancer, and their responses to standard of care drugs. So here you can see how the one well of 384 well plate looks like and how many tumor clusters we have per well. There are quite many, but also other cells present. And when we treat the samples with different drugs, we see a decrease in viability, which is apparent by the stainings. So our standard stainings are for DAPI and actin for cytoskeleton, and you immediately see changes in the number, the size of the clusters, and also their actin staining, which indicates drop in viability. So our image analysis platform is able to analyze this and convert it into numerical data, where we would analyze total tumoroid size upon treatment with different drugs and evaluate sensitivity of these patient samples to different standard of care drugs. But the most interesting value of this platform is the ability to test immuno-oncology treatments in native tumor microenvironment. And on this slide, you can see a heat map showing about 25 non-small cell lung cancer tissues that were profiled on their responses to different immuno-oncology drugs and control compounds. So our main control compound is SEA, which is superantigen. And if we see responses to this compound, we know that the sample contains immune cells, even without additional biomarker characterization. It's also apparent from the images that we take. So you can see here for this sample, the clusters are present in a non-treated sample. The control that was treated with SEA shows a decrease in the tumor cluster fraction and massive increase in single cells, indicating immune cell proliferation. And the same effect we also see in checkpoint inhibitor treatment, notably here with ipilimumab. So our platform shows that about 20% of non-small cell lung cancer tissues respond to immune checkpoint inhibitor treatments, which is also corresponding to the clinical response rate to these drugs. So this high-content imaging platform is able to distinguish decrease in the tumor cluster population compared to increase in immune cell counts or immune cell proliferation purely based on size and shape of these clusters. And this is the technology that we developed here at CBNL. So the high-content image analysis method that we developed, that I showed in the previous slide, that allows us to distinguish tumor clusters from proliferating immune cells is represented here as an example data set. You can see that in this sample, we see a considerable decrease in total tumoroid area upon treatment with SEA, very strong killing with staurosporin, and a slight decrease in tumoroid area upon treatment with beads or pembrolizumab. In addition, we see very strong increase in single cell counts upon treatment with SEA and CD3, CD28 beads, but not with other treatments. And the fact that this killing that we observe of the tumor and the increase in the single cell counts is really immune-mediated cell death is confirmed by cytokine measurements from the supernatants of the plates from this experiment. And here we did the measurements at 3 different time points. So 2 days, 4 days, and 6 days after treatment. And the high-content imaging was done at the 6 days after treatment. And we could see a very strong increase in interferon gamma, release, granzyme B, and IL-2 upon treatment with SEA, but also upon treatment with pembrolizumab, showing that the responses that we see by high-content imaging can be confirmed also by other modalities such as cytokine measurements. This is another nice example of a responsive sample that was responding to various immune checkpoint inhibitor treatments. As you can see here, it responded strongly to the killing by immunostimulatory controls, but also by the treatment by CTLA-4 and PD-1 inhibitors and CSF1R inhibitor. And the histology analysis of this original sample also showed a presence of tumor clusters, presence of immune cells, and presence of PD-L1 positive cells, again confirming our high-content imaging analysis data. So in addition to the assays that I've shown you before that employ only the cells, the immune cells that are present in the patient TME, we can also run similar assays, but with addition of autologous immune cells from the same patient. So we would receive a blood sample together with a surgery sample, isolate immune cells, typically PBMCs from this blood sample, pre-stain them with the cell tracker, and mix in a hydrogel together with tumor clusters and other cells of the TME. And this will allow us, after imaging, by image analysis, to exactly locate the positioning of these labeled immune cells in comparison to the tumor clusters and analyze immune cell migration and infiltration of these cells towards the clusters. So this is the data on autologous co-cultures in our platform that you have seen already from Maloy's presentation, so I would not elaborate a lot. But the main value of such assays is the ability to track immune cells in 3D and their relative positioning towards the tumoroids, as I mentioned in the previous slide, and really quantify the migration and infiltration of these immune cells into the cells, into the tumors, sorry. So we can not only see them by the imaging, as shown on these pictures here, but also quantify. And here for one of the samples, we have seen an increased infiltration by the treatment of SCA, but also by treatment of Maloy's compound X and the compound in combination with pembrolizumab treatment. So I hope I convinced you today about the value of this translational platform, allowing to test immuno-oncology and oncology therapies on ex vivo patient samples with preservation of native tumor microenvironment in a high throughput manner, and hopefully helping our clients to progress their lead candidate into the clinic. So this final slide is showing all available in vitro ex vivo solutions here at Crown Bio, starting with 3D tumoroids and up to patient-derived organoids and 3D ex vivo patient tissues discussed today. Thank you for your attention, and I'm looking forward to your questions.
The first question is, how does the assay take care of variability given the complexity of the TME, the distribution of immune cells, and tumor cells?
Great question. So first, we use minced tumor sample. So the tumor sample that we receive, we would use mechanical shearing to process the sample and generate tumor clusters. And during this process, also immune cells will be released from the sample. So this homogenizing process actually helps to distribute more or less equal amounts of tumor clusters and immune cells in each well in an assay in 3H4 well plate format. So in that sense, this assay offers more homogeneity in terms of TME distribution compared to, for instance, a slices assay, where you would slice the tumor sample to certain pieces, and then invariably you will have more differences in the pattern.
If I may add to Natalia's answer, after mincing, the tissue is distributed into multiple wells. So each well kind of an independent TME you can consider. And data collated from all of those wells gives a nice distribution of response. So that's one good part of the assay.
Yeah, true. Thank you, Maloy, for adding that. Indeed, because the assay is so miniaturized that we can distribute these tumor clusters and immune cells across 3T4 well plate and have 8 separate replicate wells per treatment condition. And each replicate well will have from 50 to 300 tumor clusters and even more immune cells. So it's also not a single cluster per well, but many of them. And then if you end up all these numbers, you will have quite a good representation of the TME of this tumor.
Fantastic. Thank you for your response. The next question is, how can this platform help in the development of novel drugs?
Yeah, I'll pick this up because the work that we have done, it's a novel drug. One of the biggest challenge in novel drug development is not a lot of pathways at the time of development is known. And actually, over a period of time of drug development, you know more and more involvement of different pathways, how they influence the drug's activity within human body. This particular assay, as it is taking tumor tissue directly and without any, additional modification. So this is the best where you can test your drug, which has some unknown, the closest to before you inject a human. So it kind of gives you a representation of efficacy to an extent where you know how the drug is working. And then, of course, the assay model helps and gives you opportunity to test multiple parameters. For example, cytokine release, immunohistochemistry studies, at least some amount of genomics can be done. The marker analysis, surface marker analysis can be done. So everything together, you kind of build your story on how the molecule is working. That's very helpful for a novel molecule development work.
Yeah, thank you, Maloy. If I can add to that, I think it's especially relevant for drugs in immuno-oncology space, right? Where it's very difficult to find good models that would represent the human immunity, human tumor immunity. And the mouse models, for instance, that exist out there are also quite expensive and take a long time to develop, even for humanized mice. So this model allows to test indeed in a human scenario the close the possible to the bedside of the patient and kind of put probably a final piece in a puzzle after you did all the in vitro and in vivo work on a specific molecule.
Fantastic. Thanks for your responses. The next question is, do you think there is a potential that ex vivo technology could replace animal studies?
Well, if I
please go ahead.
Thanks. We maybe we can both contribute from our own perspectives. I don't think we can fully replace animal studies at the moment. Nothing can replace a full organism, of course, and the whole interaction that happens in the body, if it's an animal or human body. As we just discussed, it can add valuable information about prediction of patient responses in this clinical scenario. But I think animal models are still very valuable for a lot of different parts of the drug development process.
So I think that obviously not at this stage, but as we are progressing more and more in these 3D ex vivo culture technologies, and as we are integrating, various imaging, genomics, molecular test, in addition to the 3D typical, the 3D, assay outcomes, we are more and more predictable from the perspective of drug function. Obviously, animal studies are used for multiple purposes when you develop a drug, correct? Efficacy is probably one of the most important one, which adds value, especially from the perspective of novel drug development or a pathway which is not fully known. we are still some distance away to replace at least some of the animal model study, not everything. I think a lot of these advancement that has been made so far on this tumoroids development, the way the 3D cells actually grow among themselves, the represents tissue in certain cases, artificial tissue regeneration, all of these technologies together, probably next 10-15 years time would be quite exciting to see at least some of the animal model study that we do now, which are predictable, but again, they have their own deficiencies. We would be able to replace, and the agencies will actually appreciate more and more this kind of data generation, especially for drug approval and evaluation.
Thank you very much for your response, Maloy. As a follow-up to the previous question, what is the potential of artificial intelligence in developing future ex vivo / 3D models?
I can comment maybe
Please go ahead
on this. Thank you, Maloy. So I think we are acquiring a lot of data. With every experiment, every patient tissue testing, the data on drug responses is accumulated, and not only in this platform, but everyone who is testing the ex vivo, and correlating this data to patient responses. And this happens in the assays where it's not only done for preclinical, but also for clinical testing is possible. And accumulating all this data together helps to build predictive models and then use these responses data to generate AI models that would be able to predict in the future, saying that if we tested, for instance, one molecule on a specific patient sample, we can predict how the patient will respond not only to that molecule, but also to the whole class of similar molecules that are developed, for instance, in the same, and for the same target or the same pathway. So in that sense, I think there is a big future also for the AI development in this space.
Yeah, my take on this particular question is, it has tremendous potential. See, the cancer, if we just focus for some time on cancer and the 3D assays that we are talking about, it's so heterogeneous. The same cancer in two different patients behave completely differently. And we have so much of evidences that, cancer is quite a personalized disease. And so it is more difficult to predict any particular treatment. For that matter, even very well-studied biomarkers also do not represent in totality the response of the drug. So data sets coming out from these in vivo model or ex vivo models has tremendous potential, because the heterogeneity between patients, even if from the same cancer type, if it can be tested very quickly, like this study is an output has 6 days, correct? And you integrate with data from this study, integrate data from genomics, transcriptomics, other biomarkers, biochemical tests, everything together. And that's where the AI models are very important. And come up with a scoring platform. So there are few companies that I have heard of are working on this kind of modeling, where a predictive or a prognostic model can be developed for particular patient, not particular disease, for particular patient. So it is personalized now. And I'm sure in the next few years' time, some of those models will be in field that will be used in trials, and the efficacy or efficiency of treatment, not really the drug, the treatment as a whole for a particular patient, especially in very cost-intensive treatments like in cancer that we see, it will be immensely benefited. And 3D, cultures or any kind of ex vivo data would be helpful in that perspective. If you look at cancer response, even the most advanced drugs in cancer, their response rate within a particular patient population is not very high. Where it works perfectly, correct? But where it doesn't work, there is a large number of patients actually who unfortunately don't get the benefit of the treatment. So the modeling studies that are going on and the integrating data from multiple sources, what ultimately it will provide a personalized treatment to that particular patient, and where there is maximum benefit from a particular drug treatment, continuity of treatment, shifting from one treatment to another treatment, combination of two different treatments. All of these decision-making process would be much more robust and helpful for the patient.
Once again, Maloy, thank you very much for your response. The next question is essentially two parts, but can we combine 3D ex vivo studies and biomarkers and genetic analysis and actually other PD markers for developing prognostic models for novel drugs in cancer immunotherapy? And what are your thoughts on that potential?
Definitely. We definitely can combine, and we need to do that. That's what we are doing already in EVPT. So we are offering already FACS analysis, for instance, combined with the functional testing. So what we presented to you today, efficacy testing combined with the FACS analysis of the tumor itself to learn on the immune landscape of the tumor. We can do genetic analysis of the tumor. We can do immunohistochemistry. We can measure cytokine responses from the functional assay. So this all adds up together, to analyze the efficacy of a specific drug. Because as we already mentioned, and Maloy emphasized that there is no single biomarker that can predict the response. It's a very complex environment, and we need to use all the possible modalities we have at hand to work with the samples that we receive, and also to try to get as much information as possible to build those predictive models.
Correct. So, as we are working on a new drug, a novel drug molecule, and fairly less understood pathway, a type of immune cell which is not yet been used in immunotherapy so much, for example, the natural killer cells. So we are very, very interested how we can develop a predictive model like that, where we integrate data from this kind of ex vivo studies and real-time data from the patients, their biopsies, the location of different immune cell, in the special transcriptomics type of study. So all of these things together actually provides confidence to the clinician that, well, this is the patient. If we select based on certain set of these criteria, we have the best possible chance to get a response. And in immunotherapy, one thing which scientists have understood for a long time, selection of patient is so critical in any of these drugs, and it is difficult, but it is very critical, correct, to get the efficacy of that particular modality of the drug, which is true for us as well, which is true for the CAR T therapy or any of the cell therapy that we are talking about today, or any of the immunotherapy that we do. So seemingly the similar kind of disease do not respond equivalently. So that's the biggest black box, I would say, in cancer treatment today with all this newer technology. I think 3D cell culture, ex vivo studies, and data from that, as Nataliia was suggesting, multiple data points can come out of these experiments, integrating them together, providing a kind of an modeling plus scoring platform, which allows you to screen patients, and that is so critical for treatment benefits for the patients.
Thank you very much for your response. The next question: how did the 3D ex vivo patient tissue platform data help you with the decision-making for IND filing of Compound X?
Okay, so for us, as and when we were developing the product, we had a unique challenge, because this molecule that we are targeting, it's the human antigen, and any of the other disease model which are available are not same. They don't function in a same way, and many of the immunotherapy targets are like that. So essentially, you have to go to a very artificial system, which in our case, we have used an humanized mice model, where the immune system is partially replaced, but it is not perfect, correct? A lot of things can go wrong, and whatever we are predicting as a response, it is not possible to set very specific controls in that experiment. A lot of assumptions are there. We tried our best to, relate to the efficacy to the drug and different immune cell activation signal, so on and so forth. So in that scenario, any regulator will try to know if this is a new molecule, how it is working, how it is functioning in human body. What is the best predictor of its function? So in that perspective, the ex vivo model, because this is completely human, we have collected the tissue. In our case, we have also collected the immune cell from the same patient. And then we are testing them together in presence of our drug at different concentration, either monotherapy or in combination. And we have the opportunity to not only measure the effect on those tumoroids, but we can analyze the tumoroids further through other technologies. So this was super helpful when we presented this data during our IND filing. There was, this gives us a strong basis that this is a particular pathway which, if can be utilized properly, and if the drug safety and toxicity can be controlled and properly estimated, could bring value to the patients who are not responding to the standard of care therapies, correct? So that's where this experiment actually helped us a lot to convince the regulators to allow us to go forward with not only monotherapy, but with combination therapy, where we can, this drug, in addition to other immune pathways, if we activate in the patients together, what is the outcome? So that way, this is a tremendous data set for us to convince the regulators.
Fantastic. Thank you very much. The next question: the 3D ex vivo patient tissue platform is extensively used in immune oncology drug testing due to TME preservation in the assay. Do you feel it can also be relevant for direct tumor targeting, such as ADCs or targeted therapies?
Well, I can maybe start, and Maloy, if you would like to complement my response, would be great. that's what we see from the studies we run, that most of the molecules we're testing are related to immuno-oncology treatments. But I think there is also huge potential for targeted therapies. Of course, it is sometimes more difficult because we don't know the target expression in advance, so these are not mechanistic studies where we would have a model that we know for sure is high expressing the target and the model that is low expressing the target, and then we're just testing for efficacy. In that case, it's better to do a study with patient-derived organoids in 3D, for instance, because those are fully characterized on their target expression profile. But this platform allows sort of more a field testing of this kind of molecules. If we know, for instance, from clinical practice that 10% of patients express the target, and then we tested the compound in our platform and we see 10% of patient tissues responding, then this is indirect confirmation. And then after the testing, we can always sequence or do any other target analysis and confirm those responses. But I think it opens up an interesting possibility, to test targeted therapies and ADCs in a more clinically related context.
Yeah, in addition to that, ADCs are, current days ADCs are quite complex, correct? And then their functionality, you're providing a specificity through the antibody, and then the functionality of the ADC, the chemical portion of the drug is quite, we are trying to make it more and more specific. Now, think of a situation in the cancers which are quite heterogeneous, like any of the solid cancers, for example, the prostate cancers or the hard to treat solid cancers. They are quite heterogeneous. The tumor is not same, correct? Mm-hmm. And that's where I think an assay like this could be helpful. And you are actually the small tumoroids that you are creating in hundreds of them in each well, correct? There is a potential of them to behave differently across a set of experimental conditions. And what, as a drug developer, you are interested in, what is the overall effect, correct? So you are essentially trying on small micro TMEs across the tumor or across the biopsied sample, and you are measuring the effect of the drug overall, correct? In some cases, you will see it's tremendously good effect. Some cases, you will not see so effect. And the prediction from that is important, correct? Mm-hmm. To understand how the drug molecule, which is very specific in terms of recognizing its target, but when it is releasing the chemical portion of that is quite non-specific, correct? That's where we will benefit more, I believe, than any animal model where the tumor is a solid tumor, has a completely different complexity compared to the human tumor as such. If a model is developed where you replicate the human tumor totally, then it's a different issue. But in most of the animal models, the tumors are very different than the human tumor occurring within our body. So I believe wherever the tumors are heterogeneous, this model will provide a better understanding of the function of the drug molecule, overall function of the drug molecule. And I'm sure the ADC developers will try this model or similar 3D models and get better data set than a more, I would say, from animal model, whatever you're predicting, which is kind of an artificial system.
Mm-hmm. Thanks so much for your answers to all the questions today. That concludes the webinar. We would once again like to thank our distinguished guest, Dr. Ghosh from Zumutor, for joining us. And thank you for staying with us for the session. Feel free to connect with us directly or visit the Contact Us page on crownbio.com. We look forward to having you join us for another webinar in the very near future.